Well, let's go ahead and get started then. It's really my pleasure to introduce Laura Johnson for her master's defense. So Laura actually did her undergrad at the U of O, graduated summa cum laude, and then went to work for Oregon Department of Fish and Wildlife as a biologist for a number of years before coming back to us to do her master's. And I have to say, Laura's my first master's student, and she has set the bar. So she joined the lab and she hit the ground running, where she was really interested in freshwater mussel dynamics. By the end of this thesis, you also will be very interested in them, but a lot of people don't know to be. She came, she was very enthusiastic about them, and the way that Laura approached her research was she went full on in the literature review, really thinking about the concepts, but then she started contacting the authors of every paper that she liked and saying, hey, what do you think would be really the important next step? Are you able to do that? If you're not, what would you advise me to do? And she was so professional and smart and enthusiastic about it that she got a lot of people on board. So Pat and I are her official mentors, but she's got this community that was really informing her work. And I was really impressed by that. And I was also really excited by the research that she was then proposing, because it was both very interesting and very important, and was identified by really experts in the field as something that needed to be done. That same year, she set about very industriously getting funding. I tried to count up how many grants she got during her first year, and then I decided to stop because I was going to miss them. So let's just say it was a lot. She was very, very diligent about this and really good at communicating why her research was so important, which I think contributed to her grant success. And then she put together, a really intense field season, and then a high quality body of work that resulted out of it. I think I would be remiss if I didn't also highlight just what a great community member Laura has been. In starting a lab, it was really important to me that I had, folks with a culture of supporting one another, of being as diligent with one another's work as they are with their own. And Laura really set the model that has created a whole group that supports one another. So she reads other people's work with an eye for detail. She gives great feedback. She supports the group as much as she puts that dedication into her own work. And I think this comes through really strongly when you look at her teaching profile. So she's by far one of the best student teachers that I have encountered, if not the best. And you don't have to take my word for it. She recently won the ENVS Teaching Award for graduate student instructors, and she had 20 people nominate her for that award because her enthusiasm and the kind of standard that she holds herself for teaching in terms of being on it in the material she conveys, being enthusiastic in how she does it, and being empathetic with students and able to kind of meet them where they are. So basically, Laura's the whole package, and I'm so excited for her to be sharing her research with you today. Over to you, Laura.
Wow, thank you, Lauren. That is definitely very, very good introduction. And I just want to confirm, can everybody hear me right now? Yes. Can you hear me, Lauren? Okay. Okay, everybody, let's get started, friends, because there's a task ahead of us, which is to cram both a beginner's crash course in what freshwater mussels are and the role they play in aquatic ecosystems, and then combine that with my research and the findings that I did, my takeaways. And I have about 30 minutes, and that just seems like a woefully insufficient amount of time. So let's just get going here. And let's see. So I wanted to start my presentation before I really jump into my research with just a little bit of a primer on freshwater mussel species and why they're important within aquatic ecosystems, because not everybody here today is coming from a stream ecology background. So freshwater mussels are an important component of aquatic ecosystems, and particularly when they occur in abundant populations. When you have abundant populations of freshwater mussels, they are performing a whole suite of ecosystem services that not only benefit humans, but that also benefit the entire aquatic community as a whole. And so one of the most well-known aspects that they contribute is water purification. Mussels are filter feeders, and so they're continually sucking water in from the water column. And then they remove pollutants and fine sediments in inorganic and organic particulate matter and bacteria. And they take some of that in as food, and then they spit that water back out essentially into the water column in a much cleaner form than it came in. So this is really well emphasized. I've got these screen captures from a YouTube video. It was a time lapse of 3 hours, 2 tanks, one with mussels, one without. And you can see that after about 3 hours, the tank that had mussels, you can see the trees starting to come through in the background. They really are powerhouses in terms of their ability to filter and purify water. And that is an enormous service that they provide, because anybody who works in wastewater management will tell you that this is an expensive proposition when humans have to intervene in systems to do this level of filtration themselves, or when this service is lost. They also bolster aquatic food webs when they are present in abundant populations. As I mentioned, they're filtering water in, and they're getting all of this inorganic particulate matter in with the water. And they're keeping some of that for food, and they're binding some of it together and spitting it back out essentially onto the stream bed next to them. And they call it pseudo feces. They bind it together with a mucus, and that food is actually food for a host of other smaller creatures, like especially macroinvertebrates. And so both that pseudo feces that they're binding together and depositing, but also their actual feces, both contribute substantially to increasing the amount of food available at the bottom of the food web. And so that is an important component of ecosystem, freshwater habitats overall. They facilitate nutrient cycling processes. They have a muscular foot that comes out of the bottom of their body, and they use it to dig down and embed in the stream sediments. And the movement that they do up and down, it helps to facilitate nutrient cycling between the stream sediments and the water column. And they do a host of other things that's really important. Their shells provide habitat. And so there's like a number of other things. I could give an entire 30-minute lecture on how they're important within aquatic habitats. But this is a little crash course right there for you. So something else really cool about the mussels, but also kind of complicated about them, is their reproduction. And it's exciting, and it's maybe a little frustrating if you're a researcher, because it makes it really difficult to study them. And I think contributes to the lack of general knowledge that scientists and resource managers have about these species. So as a little just, here's what's going on. The first step is for the mussels to breed. And in order to breed, most mussels have separate male and female species. And the male has to put his sperm in the water column, and then somewhere downstream, the female has to filter it in. So that seems complex in and of itself. But the next step is to have a female mussel who's taken in that sperm and become fertilized. She doesn't have like fully formed babies. They're like immature babies called glaucidia. And so these immature babies, in order for them to become mature, they have to be released from the mom, the female mussel, and like deposited onto a fish, but not just any fish. It has to be a like oftentimes species specific fish. And so this female mussel binds all her babies together, her immature babies, and she like leaks them out. And depending on the type of mussel species, they have different methods for how to accomplish this. In the picture here is Western pearl shell, and they just like bind all their immature babies together, their glaucidia, and they like make a mass that they dangle from their shell. And then that is interesting to the fish that they're trying to attract, and the fish will come up and maybe bite at it. And then this like mass of immature babies gloms onto the fish, which cue next picture, which is right there. Here's a fish with some immature babies attached to their gills. So the babies, in order to become mature mussels, they transform on their fish, and they're these parasitic hitchhikers for this stage of their life, which is pretty exciting and awesome. And then at some point, either weeks to months later, it's like not super clear, especially per species, exactly how long this happens. They flop, drop, and no, not roll, they settle. So, cheesy joke aside, this is a really complicated life cycle. It's hard to study. The juvenile mussels often remain buried in the sediment, like in the gravels and the sands, for maybe the first year to several years of their life. So they're not like available at the surface to look at. And this life cycle, and including the fact that mussels are really long-lived and have a delayed onset of maturity, make them really susceptible, including their role as filter feeders. They're constantly exposed to pollutants or sediment levels in the water column. If a mussel population gets wiped out for some reason, it can take a really long time for it to come back. And so this is a reason why they're also indicator species in aquatic habitats. When they're present in abundant populations, it's an indicator that that habitat is healthy overall. And when they're absent or declining, that is an indication that that habitat has been degraded or like abused in some way. So here we go. In the Pacific Northwest, we have 3 genera of mussels. We have the Western Ridge, we have Gonidia, Margaritifera, and we have Onodonta. Gonidia and Margaritifera, there's just one species, which is Western Ridged for Gonidia and Western Pearlshell for Margaritifera. And then the floater mussels are a little bit more complicated. There's multiple species of floater mussel in the Pacific Northwest, but it's not possible to ID them in the field. And so I'm just going to refer to the genera Onodonta when I'm referencing my findings for that reason. So facts. Western Ridge mussel, the least is known about it. And the host fish that it uses to transform its immature babies is likely some species of sculpin, but sculpin is a whole nother black hole in itself. Like most people can't even identify sculpin to species level. So figuring out which type of sculpin is being transformed upon, it's just, we don't know, essentially, like in most Western watersheds. This species lives for 30 years or more. It probably is living longer. There's some suggestion it can live 40 or 50 years. Question mark. Let's figure that out. It can grow up to 13 centimeters in length, and the Xerces Society actually just petitioned to have this species listed under the Endangered Species Act this summer. So this is like, we're really concerned about this mussel. This one in particular has had a number of really big die-offs of beds that we've been able to know happened. So man, Western Ridge mussel, cool. So Western Pearlshell is our most probably common, widely distributed species. It uses salmonids as its host fish, which include both salmon and trout. It can live for up to a century, which is remarkable. 100 years, like outlives most humans. It grows up to 13 centimeters plus in length and has a near threatened status according to the International Union for the Conservation of Nature's guidelines. And lastly, we have the floater mussel, the clade that I'm going to refer to with multiple species that have overlapping ranges. There's likely 4 distinct species. One just got booted out, kind of like moving around. There's multiple confirmed host fish for this mussel, including dace and shiners and sculpins. It lives much shorter than the other 2, probably 10 to 15 years. Depending on the species, it can grow to nearly 20 centimeters in shell length. And Onodonta thaliana is vulnerable to extinction in the Pacific Northwest. So if you're a mussel, there's some certain realities about living in a moving stream environment. And that's now in the modern world dominated not only by natural factors, but also by human factors. And by human, I say anthropogenic, that means human. Anytime that I spit out the word anthropogenic, I'm really just talking about people. So we look at mussels and we think about them and the things that impact them at multiple scales. At the patch scale, I'm talking about like centimeters to meters of distance on the stream bed. And there's certain things mussels need, like I mentioned, the sediments they embed in. They typically need sand or gravel-sized sediments, which are on the like very lower size class of what could occur in a stream. But things at this small scale that are also important are like, what's the depth of the water? What's the velocity during high or low flows? How far from the bank is it? Is there flow refuge present? And by flow refuge, this picture actually is a good example of it, because there's these really big boulders you can see in the photo, and the stream is moving towards us in terms of flow. And so all of these mussels are located right on the downstream side of a big boulder that's interrupting flow and creating less stress on the downstream side of the boulder as also on the upstream. So that's like a flow refuge. And those things are present on this small scale. They're important. Then we can go up to the next scale, which is the reach scale, which is tens to hundreds of meters. And at this scale, the gradient of the channel or the slope is important. The habitat type, is it fast or slow water? Is it a riffle, a pool, or a glide? What's the riparian condition like? Is it present at all? Is it healthy? What's the channel form? Is it a constrained or an open valley? All of these things play into the dynamics that are influencing habitat within the stream. And then at the largest scale, we can think about it at the watershed scale, which is a kilometer plus in size. And at the watershed scale, the geology of the watershed is really important. How fast is like overland flow being delivered to the stream? What is the distribution of host fish species like? What is the precipitation regime like in this watershed? So those are really big scales. And the thing that links mussels at both the patch and the reach scale is hydraulics. So there's this growing evidence in high energy gravel bed streams, typical in the Pacific Northwest, that the hydraulics of the stream are really influencing mussel distributions and abundances. And by hydraulics, I mean like shear stress and stream power, which are two really closely related variables. Shear stress is the force of the water column against the channel bed moving in the horizontal direction, and it's measured in newtons per meter squared, whereas stream power is the potential energy of that water column against the channel bed and banks for a specified length of channel, and is really like the capacity of the water to perform the work of moving sediment. So both of these are talking about in high flows, the ability of the water to move sediments downstream. And if you're a mussel who wants to stay stationary for 100 years, if you're in some gravel that's now getting moved downstream, that's problematic for you. So a first order control is this hydrology during high flows. Like, are you going to be in a place where the stream is moving you or not? And then once that is established, like you're either in a good spot or you're not, all these other things come into play. Is there the right sediment? Is the water quality good enough? Is there a host fish who's going to drop you there or be present? So it's complicated. It's complicated, mussels. Okay, land use. So as far as humans are concerned, we have done an amazing job of changing the face of the planet. And we have mostly changed it in ways that are really productive for us. It grows us food, it ranges our cattle, we live in the cities, we cut down the trees. But all of these processes, these anthropogenic land uses, they initiate processes at all of these scales in the river. And generally, anthropogenic land use tends to change the flow regime of streams and rivers. Low flows can get lower, high flows get higher. It increases sediment influx into streams and pollutants. It changes the distribution and quality of aquatic habitats. It can influence community composition, like what species are present in the first place. And indirectly, these anthropogenic land uses tend to help invasive species. Humans are a vector for species introductions, and then the conditions that land use changes within the stream kind of make things better for more tolerant generalist species, which invasive species tend to be. So it sort of creates these conditions for invasive species to go bonanza. So there's all of this information. So what do we think about it? Well, in the Pacific Northwest, at least, we haven't been thinking about it very much, it seems like. Maybe it's because literally mussels are easy to overlook. Can you find the mussels in this photograph? There they are. So maybe that's just like, they look like rocks. We're not thinking about them. But the mussels that we do know about in the Pacific Northwest, I mentioned these die-offs that have been happening. So there have been these large and enigmatic die-offs, and it's like, what's going on? All these mussels are just going kaput at the same time. And furthermore, there have been a few studies of mussel populations and looking at their demographies, and they haven't been finding any small mussels. Like this is a population from the Willamette River, right at the confluence of the Long Tom. And there's tens of thousands of Western pearl shell mussels there, and they didn't find any juveniles. And by juveniles, I'm using length as a proxy for age. There's a whole bunch to say about that. For Western pearl shell, I'm saying 3 centimeters and less represents a juvenile mussel. And for the floater mussels, the Onodonta, 2 centimeters or less. And that's a relatively conservative estimate for juveniles. So you can see there's no 3 centimeter or less mussels here. So where are the babies? And particularly with a long-lived species like Western pearl shell, if they stop reproducing, you might not know there's a problem if you're not paying attention until all of these long-lived animals die decades in the future. It's just like an aging nursing home. And that's an extinction debt. It's some past environmental degradation or harm that's caused this debt that will be paid in the future when all the animals essentially die. So that's cheery. But there have been some efforts in the Pacific Northwest to look into mussels. There's been a work group that was convened in the early 2000s that's been really important for building awareness, community, and identifying critical uncertainties that should be addressed. There's been a database in the works since the early 2000s, first started by the Confederated Tribe of the Umatilla Indian Reservation and others from museum specimen, and then expanded in 2008 by the Xerces Society through funds from the Nature Conservancy. And that was to really allow the first assessment of species statuses in the West, which just happened in 2017. Like 3 years ago, we just figured out a really preliminary idea of what the species statuses even are. And the Confederated Tribes of the Umatilla Indian Reservation in particular has been a real leader in freshwater mussel research and inquiry in the Pacific Northwest. They started their research project on mussels in the early 2000s. I think that they're really a leader in knowing that the cultural value of mussels is inherently tied to their resource value, and that mussels are just one part of a larger community, and their importance lies within the community as a whole. And they've done a really good job of getting long-term funding. And so they're able to look at this on a long time scale. Like if Western pearl shell lives for 100+ years, the monitoring timescale of an animal like that is just a lot longer than a lot of like normal resource agencies are able to get funding for, if they're looking into it at all, which largely hasn't been happening. And they have the only aquaculture lab in the West where they've been doing propagation of lamprey, Pacific lamprey, since 2012, and freshwater mussels since 2013 for use in restoration projects and restocking in areas where these species have been lost. Wow. So this sets us up. There's like not a lot of information known. When there is records of mussels in a basin, it's largely just like there was a mussel here, and that's about it. Like it's just not very extensive, and like so much is lacking in Western watersheds in terms of baseline knowledge of which species are present, what does their distribution look like, what is their abundance, are populations reproducing. So let's address that knowledge gap together in the South Umpqua River Basin. Here we go. This map is like got a lot going on. This is actually one of the products of my analyses, which is the land use analyses in the basin. The different colors are the different land use categories. Green is forest, brown is like a timber harvest, yellow is agriculture and pasture lands, the gray and the white is levels of development. And so we see the South Umpqua River is the thick line, the Cow Creek, a major tributary, is the thin line. You can see the sites that I visited, and the South Umpqua River was a really good area to do this study for a number of reasons, including the fact that there's no dams on the South Umpqua. So when you add the Umpqua and the South Umpqua together, it's one of the longest reaches of undammed rivers in the West. And that's important because dams cause a lot. I didn't mention that in my anthropogenic uses of rivers. Dams mess stuff up, including hydraulic and hydrologic regimes. They mess up the distribution of host fish species. So when dams are present, like impacts in distribution and abundance are likely pretty tied to that dam. But since there isn't a dam in this basin, those impacts are a lot more likely to be from these land use categories themselves. So that's why that's important. And there's a lot of gravel, the geologic zone of the South Umpqua. It's draining a lot of gravel-rich provinces. And so there's ample sediments for the mussels to embed within. Host fish are distributed throughout the entire region. So host fish limitation really isn't a problem, although declining fish abundances is another conversation. So there's like a number of reasons why this was an interesting basin. The land use is typical of a lot of watersheds in the Western US, where you've got more development in the lower river floodplain valleys, and then more silvicultural or forest uses up in the uplands. I could just keep going about it, but in the interest of time, let's talk more about why the South Umpqua is and there was sites. Like most basins only have 1 or 2 site records, and there actually was more site records for this basin. So it gave me something to go on in terms of revisiting sites that had historical mussel presence indicated. So I had 3 hypotheses. The first one was at the reach scale, so tens to hundreds of meters. I was interested in using stream power, which is something that you can calculate remotely in the lab, specific stream power, and creating a representative specific stream power for an entire reach by like averaging active channel width and a number of variables. And so you can do this remotely. And I was interested especially in the link between a reach level stream power hydraulic variable and mussel abundance, because if there was a relationship there, it could be used to identify reaches in rivers with a higher likelihood of having high mussel abundances for future surveys. Like, since so many of our watersheds are big blank slates and mussel surveys take a super long time to do, if you could like narrow that down a little bit and have a better indication of where to go, that would be super valuable, especially at that reach scale. So I hypothesized that this representative reach scale stream power is that increased for a reach, we would find lower mussel abundances. My second hypothesis was at the watershed scale, and that was anthropogenic land use. So the drainage basin that is draining to a site on the river, and this is like not only all of the waters upstream, but all of the upland, like the land and the water that cumulatively drains to that point. That's the drainage basin. And so in the drainage basin, as you have an increasing amount of percentage of anthropogenic land use, I hypothesized that not only would mussel abundances go down, but that we would see less of these juvenile mussels in particular. That might be an indication of an extinction debt phenomena. And I looked at this at 2 different watershed scales. I looked at the drainage basin as a whole, which is, you can see like all of the shaded area is the drainage basin draining to a point on the South Umpqua River, which is the site would be right where the shading and the light meet. So it's much larger, and my drainage basins ranged from like 400 square miles to like 1,400 square miles. So this is really big stuff. And then I was also interested in the US Geological Survey has identified all of these watershed units in the United States that range in size. And the smallest size that they've delineated of it's like called a sub-watershed. And it's these black sort of polygons around the river. And so this is called a Huck 12 unit. It's what the USGS calls it. And these range in size from 86 to 200 square miles. And I was interested in looking at land use at these 2 different scales to see if there was an impact. The Huck 12, the smaller scale, is definitely much more localized around the site itself, whereas the drainage basin is this cumulative impact. And then my third hypothesis was that where anthropogenic land use was intense, the influence of land use would outweigh stream power. So in zones where anthropogenic land use is a lot, that made sense, where the zones of the river that have these highest cumulative impacts from land use, you might not see the influence of stream power because land use might already be suppressing mussel abundances. And so you might not see a big difference in abundances due to these reach level hydraulics. So what did we do? We went to the stream. There were these sites that had already been identified, and we got into the river around the point and in our snorkel suits, and we started surveying for mussels. We tried to encompass both below and above each site record point. We searched for a minimum of 5 person-hours at each site. And when we found mussels, we marked their locations with a GPS point, and we initiated a timed search to like intensify efforts in that area. And at the end of that 30 minutes, we did an estimate of the approximate area of the stream that encompassed that mussel grouping. We had two different definitions that were important. One is an aggregation, which is just a group of mussels that we found that were within 10 meters of each other. And then the next thing we identified was a bed. And we said that a mussel bed, was 15 or more animals available at the stream bed surface within an aggregation. So like any grouping of mussels is an aggregation, but beds are specifically only those aggregations that have 15 or more mussels in them. So we searched all these different sites. The sites that we searched, when we did identify beds, we went back to those beds to do follow-up systematic sampling. We used a series of transects and quadrats. The white square that you see in this picture is a quadrat. It's 0.25 meters squared area. And we would systematically lay out quadrats within the beds, and then we would sample mussels at the surface. And we also double sampled with excavation to 13 centimeters depth. And we sieved substrate through a 2 millimeter mesh to find juvenile mussels. And each sampling methodology was slightly different based on the unique size and dynamics of each mussel bed. And I'm happy to talk about all that much more in follow-up questions, but I'm taking too long as it is. So we sampled, we found the mussels, we sieved the sediment, we measured the length to do proxy for age. Man, here's our facts of note. We searched 4.2 kilometers of the lower South Umpqua, 2.53 kilometers of the upper South Umpqua, and 1.38 kilometers of Cow Creek. We found 49 mussel aggregations, 15 of which were large enough to be considered beds at 13 of our sites. All three genera of Western freshwater mussels were found. And invasive Asian clams, Corbicula fluminea, were present in really high densities in the lower South Umpqua River, which is something that we did not quite expect. So we found that mussel abundances significantly declined with increasing drainage basin area. Here you can see that the sites with Asian clams have the highest drainage basin areas, and those are the sites in the lower South Umpqua River. And then as in the South Umpqua, drainage basin area in river kilometer distance are really intertwined. So as you move upstream, the drainage basin area decreases and you get way more mussels. And it's just like upstream, you could either find lots of mussels or not very many mussels. There's a lot of spread in these blue points. And then as you move downstream, it just becomes very suppressed. You don't really find a lot of mussels anywhere. And those spots without many mussels also were places where Asian clams were the most present. And here's an excavation unit. All of those yellow shells are the Asian clams. Here's a close-up of one filtering. We did some counting of clams at the lower South Umpqua bed and determined that there were almost 200 live Asian clams per quarter meter squared quadrat, which scales up in the 135 meter squared bed area to nearly 100,000 animals, live animals. There's high densities. The forest in the drainage basin area influenced mussel abundances positively. So increasing forests, you got increasing mussel abundances. And I used forest in the drainage basin as the variable of interest because it's really negatively correlated with all of the other anthropogenic land uses. And so as forest cover declines, wholesale, you are getting more of those anthropogenic land uses like urbanization and agriculture and rangelands and timber harvest. So there was a significant increase at that drainage basin level. And this was sort of a complex, this one was a little perplexing, but timber harvest within that smaller subwatershed unit encapsulating the stream. As timber harvest increased in that smaller subwatershed unit, mussel abundances also were seen to increase. And I think there's two dominant hypotheses I have about this. One is that even though that level of timber harvest in the Huck 12 was not super correlated with forest cover at the drainage basin scale, they were positively correlated. So more forest, more timber harvest. And all of these red dots here are from sites within the Huck 12 unit that had the highest amount of timber harvest in it. Like this Huck 12 unit was almost 50% timber harvest, and it also had some of the highest abundances of mussels anywhere. So there's either some residual lag in my model where this is a part of the basin with lots of forest, and so there's also lots of timber harvest, and that's where the mussels are. And my other hypothesis is that the timber harvest, the way that I classified it, it represents really recent harvest on the landscape. And so there's also the potential, since these are largely Western pearl shell mussel beds, that the harvest happened recently enough that it actually hasn't caused changes yet in the mussel populations, because there's often a pretty long lag effect between some types of land use change and what you would see in the populations in stream. And we found that even though stream power at that reach level scale, which we calculated using the 10-year peak discharge for each site, it was significant, and the model itself had a better fit when stream power was included with it, but the ecological relevance of the variable wasn't super, it wasn't super ecologically relevant. You can see here the regression line that's the line of best fit is pretty flat. And so we found high abundances of mussels at all of these different, we found abundant mussels in reaches with a range of stream power values. I think particularly interesting here is the lower South Umpqua, which are those sites with really high drainage basin areas, low in the system, with a lot of cumulative land use change. They have wholesale much lower stream power values. And the highest abundances of mussels were located in these really low stream power reaches. And so something that I've been thinking about here is that maybe the reach scale variable that I found is really only good at explaining variation when hydraulic conditions are conducive to mussels within the entire reach. And that these two dots that I've highlighted were really high stream power sites with high abundances of mussels. And it was really clear. The picture of the mussels in the boulder in the bottom right is actually an example from one of those high stream power beds. All of the mussels were only found immediately downstream from flow refuges. And I didn't do a good job of measuring this in my study, but it's possible that that hydraulic factor is influencing beds at different scales. And so my reach level representative value isn't able to pick up this patch level variation. And so it's something that I'm thinking more about and wanted to mention. In terms of my bed demographies, I surveyed 3 Western pearl shell beds and 1 floater mussel bed on Odonta species. I found reproduction in all of my Western pearl shell beds. The red hump is the lower South Umpqua River, the blue hump is the bed in the upper South Umpqua River, and the green hump is the bed in Cow Creek, the large tributary. The blue bed in the upper South Umpqua River had a really high density, like 90 mussels per meter squared. It was skewed towards older individuals. And this is really indicative of a bed in a hydraulically stable zone where the mussels are able to live to a really long age. And there can be really high competition for space in these beds. And there can actually be a suppression of juveniles because the adults are so long lived. And so this blue bed in the upper South Umpqua is, even though there's a low number of juveniles overall as a percentage of the population, all of the size classes are represented, and it's likely a viable bed in terms of being able to sustain itself. And if that's true, it's a really important source population within the river. The bed in Cow Creek was much younger skewed in general, and that's that green blob. And bed distribution like this is really typical for populations that are periodically scoured. And so none of the individuals are really able to reach a high age, but there is reproduction happening. And then this red bed in the lower South Umpqua, even though individuals are present from all different age groups, including the juvenile mussels, only 8 animals came out of our sampling overall. And so it's the one bed that I didn't hit my minimum number of sampling requirement. Like I'd really wanted to sample 50 animals to do a successful bed demography. And so there's just like low abundance in the South Umpqua River overall. Although juvenile mussels were present, but Western pearl shell can be hermaphroditic at low densities. It's sort of a unique mussel for that reason. And so it's possible that these low densities in the South Umpqua are resulting in selfing of hermaphroditic, like females just are able to fertilize themselves. And that has really important implications for like genetic viability of this species in that zone, if that is how reproduction is happening in those areas. Wholesale, I was able to, and then the one other demography that I did was for floater mussels. And there were a lot of juveniles in that bed. There were two, it was bimodal, so there were two big peaks in the size class of mussels there. And it's probably typical for a population that's having like pulses of reproduction. In wholesale, especially in the floater mussel bed, there was a burial rate of 4. So every one mussel at the surface, there was 4 mussels buried in the sediment. And wholesale, it was juveniles that were buried. And so excavation was really important to include, especially for that floater mussel bed. The Western pearl shell beds, oh, and there's a juvenile mussel right there. That's a floater mussel for you. That is like 0.6 millimeters in length. They were very tiny. In the Western pearl shell beds, it was important to have some excavation. This is for Cow Creek. We got 1 mussel out of our excavation unit. It was a juvenile. For our bed out in the upper South Umpqua, we pulled out 10 juveniles in our excavation units. They tended to be juveniles overall, but they weren't a huge part of the population. The burial rates for our Western pearl shell beds were much closer to 1, and we could have done less excavation in order to adequately capture that component of the population. So let's summarize, shall we? Because I'm already over time. Thank you for sticking with me. I think that we need to be systematically surveying more populations in the upper river. There are at least 3 beds that I found that I didn't get out to survey. And the beds that we have already surveyed, we should be doing repeat monitoring. You need to be able to get out population growth rates and mortality rates to be able to start moving towards doing population viability analysis or minimum viability analysis to really be able to like predict what these populations are going to do into the future and manage for that. So we just really need to keep working on getting more information, including more site assessments. All of my sites were actually clustered within 1 geologic province. And so I didn't survey in the super far downstream or for super far upstream parts of the river. And we should be looking there too. And I think that my results suggest there might be some potential for using these like representative hydraulic variables to direct future surveys. We just need to increase sample size, and that needs more investigation. And we really need to be understanding what impact these Asian clams are having on native mussels, since their ranges are set to increase in the future. And I think that it would be nice to establish an age-length relationship for the Umpqua Basin in particular, because these have been established for other basins, but growth rates are different depending on where you are. And so doing that to be able to measure animals and then say something about how old they actually are would be beneficial. My lessons learned, just because you can doesn't mean you should. Interjecting within the system inevitably changes it. And I think that I learned some beds shouldn't be sampled extensively due to these patch scale dynamics. Like those beds in the high stream power reaches where mussels are just found in this fine sediment that's able to accumulate behind large boulders. By trying to sample that bed, you would destroy it. And so like knowing that we need to have this information, but we need to be thinking critically and ethically about what's the best way to get it. Ask questions, admit when you don't know, and keep detailed notes so you can improve for next time. And lastly, do everything you can to ensure the information you find is accessible to the community for which it's relevant, which I think can actually be really hard to do sometimes, because you get your work done and you find meaning and value, and it's a whole nother step to try to spread that word. And it's part of the reason I'm grateful so many of you are listening to me here today. It's definitely more people than I had felt initially comfortable with attending my defense. I want to acknowledge the Environmental Studies program at the U of O. They provided me funding through their Barker funds. and support through their awesome people community. The Oregon Department of Fish and Wildlife donated a bunch of gear to use for these projects, and turns out renting a snorkel suit is super expensive. So probably could not have done any of this without the Department of Fish and Wildlife donating that gear. The Northwest Ecological Research Institute provided important grant money. The Xerces Society for Invertebrate Conservation, and in particular, Emily Blevins, she took me out and taught me how to survey for mussels and has been a huge mentor and a source of information for me in this journey. Thank you to the mussels themselves. They are powerhouse worker, awesome superstar, diva, fabulous star, wonderful. It's the mussels. Thank you to my field assistants, Kennedy Potts, Emily McCoy, April Dawn Christian, Jamina Shupak, and Michaela Fishback. I didn't pay any of you enough money for the work that you helped me to do, so hopefully I compensated you in knowledge and fun times on the river. Thank you to the Hallett Lab members. So many of you now, because the lab has grown, but in particular, Ashley, Alejandro, Lena, and Elisa, thank you so much for reviewing my work. You've really made it a better body of work in the end. And with that, I will stop talking. 10 minutes over, dang.